Part 2. Cellular Respiration. When we left off, the CO2 warden had made Henry the hydrogen do his evil bidding and create one glucose molecule. To make sure Henry didn't talk to anyone, the warden decides to put him in the mitochondria, also known as the maximum security prison. So Henry enters the mitochondria. Pause. Given the situation, the warden decides it'd be safest to split his money in two. So he takes 2 ATP and splits his glucose by removing 1 pyruvate, making ADP, into 2 pyruvate. During this process, 4 hydrogens and 4 electrons are released as well. This is known as glycolysis. 4 ATP are also produced. To prevent any prisoners from getting his 2 pyruvates, the warden decides he's going to enlist the help of NAD+. The NAD+ guard comes across 2 electrons and 2 hydrogens. He captures 2 of the electrons and 1 of the hydrogens, but 1 of them gets away. Then he goes off to another part of the maximum security prison. The warden decides he has an idea of how to get Henry the hydrogen back, baiting him with his money. He converts each pyruvate molecule into acetyl-CoA, which he then sends into the mitochondria, into the Krebs cycle, aka the bait and wait. The warden and acetyl-CoA enter the mitochondria, and the Krebs cycle begins. Step 1. First, the warden and his acetyl-CoA are transformed. The acetyl-CoA combines with oxaloacetate to form citrate. Step 2. The citrate then becomes isocitrate, and H2O is released. Step 3. Isocitrate is then transferred to alpha-ketoglutamate, and the compound gives off 1 hydrogen ion, which is then picked up by NAD+ and taken off again to another part of the mitochondria. At this point also some CO2 is released. Step 4. Alpha-ketoglutamate is then transformed into succinyl-CoA, at which point the warden decides he's had far enough and leaves the cycle, leaving his money. Step 5. Succinyl-CoA is transferred into succinate, at which point 1 ADP molecule and 1 inorganic phosphate are charged enough to produce 1 ATP. Step 6. The succinate is then transformed into fumarate, giving off 2 hydrogen ions, which are picked up by another electron carrier, FADH. It picks them up, becoming FADH2, and goes off to another part of the mitochondria. Okay. Step 7. Fumarate is transformed into malate, once again giving off H2O. Step 8. Malate is transformed into oxaloacetate, the beginning part of the cycle, but 2 hydrogen ions are given off, leaving a very empty bag of money, ready to be recharged by more, because there are 2 pyruvate for every 2 acetyl-CoA for every glucose made. To pick up these hydrogen ions comes our good friend, but not really, NAD+. Again, taking them away to another part of the mitochondria. So in the Krebs cycle, or the bait and wait process, 1 ATP, 1 FADH, and 2 NADH are sent over the matrix. The final step is oxidative phosphorylation, Henry's great escape. The NADH and FADH2 guards escort the hydrogen ions and the electrons into the inner part of the mitochondria, known as the matrix. So the electron carriers escort them to a sort of rehab center inside of the mitochondria, known as the electron transport chain. It is a series of protein complexes as well as more electron carriers and cytochromes, which with iron hemes in them, they attract the electrons in the chain. This process begins when NADH ferries the two hydrogens and electrons to protein complex 1. At this point, the electrons go across the protein complex, and the hydrogens are sent out of the ETC and into the intramembranous space via an exergonic reaction from the electrons. Next, FADH2 drops off its electrons and hydrogens at the protein complex 2. Again, the electrons are released into the ETC, and the hydrogens are ferried across the membrane into the intramembranous space. The electrons then board ubiquinon, an electron carrier, taking it over to protein complex 3. Next, cytochrome C picks up those electrons, bringing them to protein complex 4, the final step of the chain. Because they want to follow the electrons, some loose hydrogens also go through the protein complexes into the intramembranous space. At the end of the chain is the most powerful electron acceptor of them all, the power-hungry oxygen. The electrons go through to meet him, but now there's a problem. All the hydrogens on the other side of the membrane desperately want to get back to their electrons, and there's only one way through. So Henry finds his way to the ATP synthase, but secretly, he's going to make his escape. By going through the final turn of the ATP synthase, the laundry room, he will be reunited with his electron at last. And here's how it works. With the great ion concentration gradient on the outside of the cell, it's no problem for the hydrogen ions to go through ATP synthase. With every turn of the synthase, however, one ADP and one inorganic phosphate are combined to make ATP. So all the hydrogens go through, each creating its own ATP. Finally, it is on the interior that Henry the hydrogen and his electron are finally reunited in the strangest way possible. By teaming up with the oxygen atom, they form H2O. Finally reunited, they make their escape from the cell in the form of sweat glands in the human body. Once again, Henry and his electron are reunited, and the warden, whose evil plan failed so poorly, is left unhappy. So, with the 2 net ATP from glycolysis, the 2 ATP from the Krebs cycle, and around the 34 ATP from the oxidative phosphorylation, we gain a grand total of 36 to 38 ATP in the process of cellular respiration.